So let's get started. My name is Julie Wong. I am representing the Alzheimer Society of Toronto. This Advances in Dementia Research series is actually presented by the Toronto Dementia Research Alliance in partnership with the Alzheimer Society of Toronto. And this series discusses recent updates in dementia research and highlights dementia research studies occurring across Toronto. Today, our speaker will be presenting on updates on non-invasive brain stimulation in Alzheimer's disease and related dementias. So a little bit about the Alzheimer's Society of Toronto, in case you are not familiar. The Alzheimer's Society of Toronto provides free education, counseling, programming, and information to persons living with dementia, their care partners, whether it be family members, or friends. We also offer training to healthcare professionals as well as general public. Something that we do always want to stress is that you do not need a diagnosis in order to seek support and services from the Alzheimer's Society, and we service all dementias, not just the Alzheimer's disease, even though that is our name. Some information we have in terms of our contact, and as well as our online referral form. You can self-refer, or others can refer you, so long as consent of whoever is receiving the services is obtained. We also do have our education platform on AlzEducate, and so this is a space where you can do self-paced courses as well as access webinars that we have, whether it's recordings or live webinars. And to see all of the events that we have occurring at the Alzheimer's Society, we've also included the listing there. I'd also like to tell you a little bit about our partner, the Toronto Dementia Research Alliance. So the Toronto Dementia Research Alliance was established in 2012 and is a collaboration amongst the University of Toronto and affiliated academic hospitals, including Baycrest, the Centre for Addiction and Mental Health, Ontario Shore Centre for Mental Health Services, Sunnybrook Health Sciences Centre, and the University Health Network. TDRA is working together to better understand, prevent, and treat dementia by creating a stronger link between basic science and clinical research, embedding research into care, improving outreach and education to the community, and increasing the efficiency of collaborative research processes across the city. Before I hand it off to Natalie, I do want to let you know that the webinar is being recorded and will be accessible on the TDRA website, as well as the Alzheimer's Society of Toronto YouTube channel. And so all of our webinars are posted there. All right, it's all yours, Natalie.
Okay, great. If we could just move to the next slide. Hi, everyone. My name is Natalie Dren. I am a research coordinator at the Toronto Dementia Research Alliance. And the basis of these webinars is promoting research that we have listed on the Toronto Dementia Network. So AST and TDRA have partnered to try to make research more accessible to everyone. The Toronto Dementia Network, or the TDN, is a website that lists dementia-related programs and services across Toronto. And on this website, we've now created a section called Research Studies, which lists dementia studies that are open for participation across the City of Toronto. Each study listed is led by TDRA-affiliated scientists and has been approved by something called a Research Ethics Board. There are studies for people with Alzheimer's disease and other types of dementia, mild cognitive impairment, those at risk of developing dementia, healthy volunteers, and caregivers, amongst other groups. The studies that are listed are all summarized in plain language. And if you would like to get started in looking at the website, the link is shown on the slide here in orange. Information about the TDN can also be found on both the Alzheimer's Society of Toronto website and on the TDRA website. Next slide. On the website, there are two main pathways. So first, you can browse for a study using search terms, filters, and quick searches. When you find a study that you are interested in, you can fill out the contact form to be connected with the research team who will enroll you in the study if it's something you're eligible for. Or at the bottom pathway here, if you're not sure where to start, you can fill out a questionnaire to be matched to a study. And any information you enter on any of the forms on the website will be securely stored and protected. Next slide. Okay, so this is what the website looks like. If you just search Toronto Dementia Network and not research studies specifically, you can get to the research studies section by clicking the tab along the top right, which is circled here in red. When you're on the page, if you're looking for something specific, so for example, Alzheimer's disease, or you know the study name you're looking for, you can type it in the search bar along the top. You can then either hit submit with that term, or you can apply more filters like the type of study, age group, target population, or the study location, and then hit submit. Alternatively, you can use the quick search buttons we have in the blue boxes, or you can search using filters without using a search term. If you'd rather fill out a questionnaire to be matched to a study, click the Match Me to a Study quick search box, or scroll to the bottom of the page and you'll see this orange button that says Can't Find a Study You're Interested in, which will also take you to the questionnaire. Each study summary, as shown in the PAS-MCI example here on the left, describes what the study is about, who can participate, the location, and the time requirement. And again, this is all in plain language. PAS-MCI is the study that we will be discussing today, so you can check out the listing after the talk, and if you're interested in participating or learning more, all that information will be on the TDN listing. Next slide. And now I'd like to introduce our speaker. Before I do, just a reminder to keep any questions you might have till the end of the webinar for our question and answer period, and we will try to get to all of them then. So today we're grateful to be joined by Dr. Tariq Raji. Dr. Raji is a professor of psychiatry and director of the Geriatric Psychiatry Division at the University of Toronto and the executive director of TRA. He is also the chief of the Adult Neurodevelopment and Geriatric Psychiatry Division at the Center for Addiction and Mental Health, or CAMH, and a clinician scientist in CAMH's Campbell Family Mental Health Research Institute. Additionally, Dr. Raji holds a Canada Research Chair in Neurostimulation for Cognitive Disorders. So welcome, Dr. Raji, and we look forward to hearing about non-invasive brain stimulation and the PAS-MCI study.
Hello, everyone. Thank you, Julie. Thank you, Natalie. Thanks, everyone, for joining. I'm going to share my slides. Do they look okay?
Yeah.
Okay, great. Thanks. So yeah, it's really exciting to present that. It is it's different than my typical presentations that which I'm really excited to share with you. And I, on purpose, as I was mentioning to Julie and Natalie, on purpose I have the presentation relatively short to allow enough time for discussion and questions about brain stimulation, non-invasive brain stimulation in the study itself, given that it's a high technical field, and there may be some questions that to clarify some aspect of the techniques that I'll be talking about and being used. Before I start, I'd like to show you the different funding agencies and support that my lab receives, myself and the team members, and specifically for the PAS-MCI, it's been mainly funded by CHR, Canada Research Chair, the Canada Foundation for Innovation, and some of the work you see has been funded also by the Western Brain Institute. These are also some relationship with potential conflict. Nothing really, there's nothing conflicted here, especially to this presentation. So I think what I thought to start with the presentation is going back to what is the scientific basis for using what we think is for using brain stimulation, non-invasive brain stimulation in Alzheimer disease or Alzheimer dementia. Can you hear me while I'm getting a message? You can, okay. And it really goes back, in my opinion, to the idea of neuroplasticity and brain plasticity. And here, what you see is the photo of Donald Hebb, who's a Canadian, and who was really one of the pioneers, one of the first people who thought and conceptualized the idea of neuroplasticity and brain plasticity. And some of the quotes we often use is that neurons who fire together get stronger together in some. That's one approach to look at neuroplasticity. But what you see also here are notes from his early work, and again, this is conceptual, theoretical work that is pointing to the fact that if two cells, two neurons, are connected and fire together and are strengthened by firing together, then the connection between them becomes stronger. And that was proposed as one of the biological bases for learning and memory, how the network learns information. And this is to illustrate this a little bit more. So what you see here in the top and under A is, let's say, a neuron that is getting an impulse, a stimulus, but that stimulus is not enough to generate activity, to activate that neuron, which is represented here by like a star-shaped like neuron. But when that same neuron receives high frequency stimulation, high input, then the activity becomes strengthened. It gets potentiated. The response of that neuron to the same weaker response, which is in panel C, becomes stronger. So what's happening is that the thinking here is that this high frequency stimulation strengthen the ability of this neuron to respond to a weak input, which before that high frequency stimulation in the panel A, that weak input did not activate that neuron, that brain cell. So one way to think about this is that it's a mechanism that the brain uses so that it learns to respond to weaker inputs and forms a memory of these weaker inputs by combining them with high frequency stimulation and inputs to these neurons. Now, this was, and I'll walk you through the slide. This was work, Donald Hebb and others back in the beginning of the century, were theoretical work that proposed how the brain learns, how the neurons connect with each other to form memories. And it wasn't until the 70s that theory, that hypothesis was shown to be to have evidence in the experiment in the lab, by having experimental evidence with the discovery of what we call LTP, or long-term potentiation. And what you're seeing here is this traditional classical setup for LTP, where you see in the box, in the square, and the rectangle, a slice of the hippocampus from preclinical animal models, typically rats or mice. And the hippocampus is a region of the brain that's highly involved in memory, in forming new memories and remembering information. And what the discovery of the LTP consisted of is that if we look now at this graph before the word tetanus, what you see is that stimulation, so these are stimulation to that neuron and the hippocampus, that's not resulting in significant activity. So the activity, what you see is the level of activity is at baseline, and it's reflected by, a score here of around 0.02 of EPSP. That's the measure of this neuronal activity. But then when the tetanus is delivered to this slice, to this neuronal section, that high frequency stimulation results in that weak input causing higher activity and higher response from after the high frequency stimulation. That high response is what we refer to as LTP, or long-term potentiation. So that synapse that was subjected to this high frequency stimulation learned to respond stronger to the weaker input, to which that synapse was not responded strong enough before the stimulus. So that was experimental evidence, lab evidence that this idea was proposed by Hebb and others at the beginning of the century is actually happens in the brain and has been linked to how the brain forms memories and learns new information. Now, with more advancement in the field of learning and memory and brain plasticity, neuroplasticity, there has been development of different protocols, different stimulation protocols that cause this potentiation, this strengthening of the synapses. Synapses are the connections between the neurons. So one form, we talked about the high frequency stimulation. So you see here like stimulation at 100 hertz or 125 hertz. So these are 125 pulses per second. Another form of stimulation is called theta burst stimulation. So these are bursts of high frequency pulses. You see them here in groups of threes that are repeated at a slower frequency. And these are, again, these are protocols, simulation protocols that have been developed and discovered in animal models and in preclinical models and slices of the brain from animal models of learning and memory. Another form of stimulation is called spike timing dependent plasticity. And here, the example here, what you're seeing is that when you have one neuron, which is the vertical one, that is connected to two other neurons. These are the horizontal ones. So the brain cells here are the neurons. So you have one vertical and two horizontal ones. The horizontal ones are connected to this vertical one. So if you have one connection that is strong, you see that leads to strong stimulation of this vertical neuron. If that strong stimulation is paired with the weak stimulation, which is what we see here as pathway 2, what you end up happening if you pair those stimulations together, the weak stimulation over time becomes strong on its own. So again, if you think about it, this connection between in pathway 2 that initially was weak becomes strengthened. So there is a new connection, and which translates to a new piece of information being learned if we believe that these connections are the biological basis of learning new information. So why did I spend time talking about these paradigms, these stimulation protocols that have been discovered and tested in slices, and so in bench work and in the labs to generate brain plasticity in slices? Because a lot of the non-invasive brain stimulation techniques that we use in humans to treat neuropsychiatric conditions or to try to enhance memory, to try to enhance cognition in patients with Alzheimer dementia or patients with MCI, mild cognitive impairment, or other forms of treatment, for example, for depression, these paradigms that use magnetic stimulation or electrical stimulations were developed based on these paradigms that were discovered for slices, for animal models. So I just wanted to share with you the basis for what led to the development of these TMS or magnetic stimulation based interventions or electrical stimulation interventions. So a common well-established common form of brain stimulation, non-invasive brain stimulation. And when we say here non-invasive, we're referring to interventions that don't require surgery, for example, and don't require anesthesia. So these are interventions that are delivered in the clinical settings, in the clinics. People are awake. Many times they are actually engage in some other form of cognitive activity, like cognitive training, memory training. So a common one is RTMS, or repetitive transcranial magnetic stimulation. So in this paradigm, this one simulates, so it mimics the high frequency stimulation protocol I described early on in slices. So what happens here is you have a magnet that delivers pulses, magnetic pulses at high frequency. Now we cannot get to the same level of high frequency of 100 hertz or 125 hertz as we've seen in the slices in the bench work because of safety. So typically we use 10 or 20 hertz and with some variations. And so what happened during magnetic stimulation, the coil, so that coil that you see here in black, it generates this magnetic field. So it has electrical current that circles within the coil, what you see here in yellow, and that circular electrical current generates magnetic field, what you see here in red, which interfaces with the brain, with the cortex, with the brain tissue, to generate what we call eddy currents, which are again electrical currents now inside the brain that activates the neurons and activates the connections between the neurons. So that is the physical mechanism through which TMS works. So using magnetic stimulation and generating magnetic fields that generates electrical currents in the brain that the brain understands. The brain understands electrical currents and activates its neurons on the surface, especially on the surface of the brain, through these electrical currents. And again, there are many ways that TMS can be delivered. RTMS, which is the traditional repetitive transcranial magnetic stimulation, delivers that high frequency stimulation. There is also a form of magnetic stimulation, or TMS, called theta burst stimulation, using the same terminology and the same approach as theta burst stimulation that we saw previously in animal slices, where instead of delivering a course of pulses, magnetic pulses, let's say at 20 hertz for a period of 30 minutes, in theta burst stimulation, or TBS, these pulses are delivered in clusters of 3 pulses at a very high frequency, separated by lower frequency and larger interval. Excuse me. The TBS has been recently approved, for example, in the US to treat depression. RTMS has been approved in general in the treatment for the treatment of depression for many years now. TBS, the advantage of it compared to traditional RTMS is that each session can be very brief. It can be around 3 to 4 minutes versus a typical 30 to 40 minutes of typical RTMS session. That's again for the treatment of depression, but there are some work looking at TBS and RTMS for the treatment of cognition in people with dementia or at risk of developing dementia. Another form of non-invasive brain stimulation uses electrical stimulation directly instead of using magnetic stimulation. So these are called transcranial electrical stimulation, or TES versus TMS. And what you're seeing here is an example of the machine that generates, in this case, a direct electrical current. So really the machine in a in a nutshell, it uses a battery to generate typically 2 milliamps current, direct current, through 2 electrodes in this case. What you see is one electrode would be an anode, which the tissue, the brain tissue under the anode experience excitation. So where you put the anode, the tissue under it gets excited, versus under the cathode, so the current, the stimulation under the cathode becomes inhibitory. So it results in inhibition of the tissue. And so the advantage of this technique is that it's relatively much cheaper to develop than TMS machines. It's also portable. So these machines, people can take them home and they can use them either themselves or with the help of caregivers or care partners to help them receive the intervention. The form of stimulation, again, like I said, is electrical, and when there are only 2 electrodes of that typical size, which tends to be let's say 3 to 5 centimeters, the stimulation tends to be broader. So the area of the brain that gets to stimulated tends to be a broad area of stimulation, compared to when we use usually TMS. Usually the stimulation is very focal in a particular region, and it's also typically on the surface of the brain when TMS is being delivered that is activated, versus when we use TES, or in this case, transcranial direct current stimulation. The current flow through the brain, so the regions, you have the surface region activated, but also you may have some deeper regions of the brain being activated as well. Now, there is another form of electrical stimulation called high definition TDCS or TES. In this case, what you see here is multiple electrodes over the scalp. And the advantage of this approach is that because these are multiple and you have anodes and cathodes, so excitation and inhibition surrounding the excitation, you can get more focal areas of stimulation versus the previous model I showed you. And you can also end up being able to stimulate multiple regions at the brain in a focal way versus using only 2 large electrodes with sponges like I showed you in the figure before. Now, when we talk about electrical stimulation, in addition to the electrodes and the types of electrodes that we're using, in addition to the size and whether it's high definition or not, we can also think of the nature of the current that will be being used in delivering stimulation. So as we know, there is AC and DC current, so direct current or alternating current, and the stimulation can be of that nature. So when we use anodal transcranial direct current stimulation, so that's the TDCS, what you see on the right side, you end up having excitation, and so you end up having one direct current goes up and then goes down. On the cathode, under the cathode, you get the inhibition. However, when we use alternating current stimulation or TACS, the current is alternating as it's termed, so it's not a direct current. And what is the advantage of that approach is that we can, with TACS, we can stimulate the brain following the oscillations of the brain. So when we record electrical activity from the using EEG, let's say, of the brain, we can detect brain oscillations, wave oscillations at different frequencies. And what we can do with the TACS is stimulate by targeting an individual specific brain oscillation at a certain frequency. So the current that we're delivering starts to resonate with the natural brain oscillation of the individual person we're delivering the stimulation to. So that's something we cannot do with direct current stimulation alone. Another form, so that it's going to take us closer to the PAS-MCI study that Natalie mentioned at the beginning of the talk. So another form of brain stimulation that uses both magnetic stimulation and electrical stimulation, but in this case, electrical stimulation of the peripheral nerve. This is called paired associative stimulation. And remember, I was explaining that many of these paradigms mimic or simulates the animal work that I showed you at the beginning. This one, it simulates or it mimics what we call spike timing dependent plasticity. So what happens in this paradigm? So first, what we do, we use the magnetic coil to stimulate a certain region of the brain. Let's say the motor area of the brain, the region of the brain, the motor cortex that activates, for example, the hand muscles. And then what we measure, what you see, so let's look at the, on the left side, when you have the coil only, and it's under the word test and pre. So what you see under that is a response, a measure from the measuring electrical activity from the hand muscle. So you measure the, you put electrodes on one of the muscles of the hand, and then you measure the electrical activity of the hand muscle in response to delivering TMS only to the motor region of the brain. Then after that, we start pairing the TMS stimulation with electrical stimulation of the median nerve. So that's a nerve at the wrist level, at the wrist of the hand. By stimulating that nerve at the wrist, it generates input that goes all the way to the brain region, that is the motor region of the brain, where we are delivering also TMS. So what's happening is you have two inputs, TMS and electrical stimulation from the nerve at the wrist level. These two inputs coincide together. And what happens after we pair these two inputs, let's say 90 times, or sometimes we do it 180 times, we deliver then TMS alone. So that's the post, and on the right side, you see test and post. We deliver now TMS alone again to the motor region, motor cortex of the brain. And what we're seeing now, if you compare the response at the lower bottom end of the figure, you see much stronger response in the hand muscle. So that strengthening of the response, that potentiation, mimics what we talked about at the beginning, long-term potentiation in the slices. And so that's one way to induce, to generate brain plasticity in humans using TMS and electrical stimulation. So what I just showed you is the a paradigm as originally developed for the motor region of the brain. Now, we're interested in neuropsychiatric conditions like dementia, like other forms of mental health conditions. We're interested in the frontal region of the brain, or the frontal cortex, or specifically here, what we refer to as the DLPFC, the dorsolateral prefrontal cortex. So that's part of the frontal region of the brain. So what we do, we in this paradigm, we follow the same idea, the same protocol, but instead of delivering TMS to the motor region of the brain, we deliver it to the frontal region of the brain. And instead of measuring activity from the hand muscle, which is connected to the motor region of the brain, we measure activity using EEG. So that's how we measure the output of the frontal region of the brain, by measuring something called CEA, or cortical evoked activity. And we use then the same approach. We use TMS alone first, then we pair TMS with peripheral nerve stimulation or PNS to the median nerve, which is at the wrist level. And then we measure the response with TMS alone after the pairing. And what we've shown when we developed this protocol is enhancement of the frontal region response to this pairing. So what you're seeing, if you look at the left side, where you see red on the right top plot here is on the PAS. So PAS is the active stimulation. PAS-C or PAS control is a control condition. What you see is enhancement of the frontal region of the brain. This is what you see in red. Also, you see it here, kind of pre and post with the bar graphs on the right side of the slide. So this slide shows the development of this approach in healthy individuals. So these were adults, healthy individuals. They did not have any brain disease, but it was showing the evidence that we can apply this paradigm in humans to the frontal region of the brain and show plasticity or enhanced plasticity in the frontal region of the brain where we stimulate. So then after that, we adapted this approach to measure brain plasticity in patients with Alzheimer dementia. So what you're seeing here is two groups of individuals. If you look at the colored figures, again, these top plots, at the bottom are patients with Alzheimer dementia or AD. These were 32 individuals. At the top are healthy controls, healthy individuals, were 16 of them matched to the individuals with Alzheimer dementia. And what you can see is that the ability of patients with Alzheimer dementia to experience plasticity or to experience this potentiation in response to this PAS protocol is weaker, you see it just by comparing the colors, weaker than in healthy individuals. So the colors are hot in the orange and red region, yellow, orange, and red region in healthy individuals, but you don't see that much response in patients with Alzheimer dementia. It's still there. You still see some potentiation, but it is weaker. So that's after delivering one session of PAS. So then we ask the question, what happens if we deliver PAS, that intervention, paired associative stimulation, in people with Alzheimer dementia, but more than once? How about we deliver it 5 days a week for 2 weeks? Can we increase the ability of the brain to experience potentiation, to experience plasticity, if we deliver this intervention 5 days a week for 2 weeks? So what you see here is the flow chart of a pilot study that we conducted. In patients with Alzheimer dementia, early Alzheimer dementia, what you see is that how many people we screened, how many people consented, how many people were excluded for different reasons, and then how many people were randomized to receive active repetitive PAS or paired associative stimulation. So that's on the left side. So that's what you see it as RPAS. Repetitive meaning it's repeated 5 days a week for 2 weeks, versus 16 other people were randomized to receive the control. So not the active intervention, but the control intervention. So then people received those, like we said, for 2 weeks. And then we did follow-up. So we assessed their brain plasticity, the frontal region plasticity, at day 1, day 7, or day 14 after the intervention, after they finished the 2-week intervention. And these were the results. So what you see here in blue are people who received the active RPAS. In green are the individuals who received the control RPAS or the sham RPAS. So I'm going to walk you through each panel one at a time. So if you look at the upper left panel, this is showing the change in brain plasticity from baseline to 1 day or 7 days or 14 days after receiving the 2-week intervention. And what you can see is that those who received the active intervention experienced enhancement in brain plasticity at the frontal cortex. That's what's represented by the asterisk here, versus those who received the sham or the control, which is represented by the green color, did not experience any change. Unfortunately, we did not see persistence of that enhancement at day 7 and day 14, and we can talk more about why we think that is the case. So there was enhancement in plasticity immediately after receiving the 2-week course in the active arm, not the sham arm. If we look at on the upper right panel, what we measured here, when we look at the frontal cortex, we're looking at something from a behavioral perspective, from cognitive perspective. We measure something called working memory, which is highly connected to the prefrontal cortex, to the frontal region of the brain. What is working memory? Working memory is this ability to hold information online and to manipulate this information. So for example, when you're doing mental arithmetics, if you're doing mental additions or subtractions, divisions, you're using your working memory because you're holding the numbers that you want to add or subtract, and then you're doing the manipulation of these numbers, the operation of subtraction or addition. So that's working memory. So what you see here is that those individuals who received the active intervention, the active PAS, also experience improvement in working memory compared to those who did not, who received the sham intervention. On the left lower panel, we also measured using EEG an index of the brain that is called theta gamma coupling. So that's we won't go into detail now about what this measure is, but this is the connection of the frequencies between. We talked earlier about the brain has its own rhythms, its own brain rhythms, brain oscillations. So that's the coupling between different oscillations of the brain, which we've shown it's linked to working memory, to link to that ability to order, to manipulate information. We also saw the improvement in this index after the paired associative stimulation, the active intervention, but we did not see that in the control arm. Then on the last panel, which is the right lower panel, you see the association, the correlation between how much improvement happened in this measure or coupling. The oscillation measures was associated with the improvement in performance on the working memory task, on the behavioral task. So all this work, this was pilot work done in 32 individuals, led to the to receiving the funding from CHR to do a large trial that is now happening, ongoing, what we call PAS-MCI, which is using the same paradigm, this combination of TMS with electrical stimulation, but in individuals who have MCI, not early dementia. And what we're doing in this case also that's different from, we're doing a few things that are different, but one of the key differences is that we're stimulating both the left and the right frontal cortex versus in the data I've shown you so far, because this was a pilot, we only stimulated the left frontal cortex in that region. So maybe by stimulating both regions, maybe we'll see more sustained response, not just immediately after the intervention. This is the setup, what you see on the right side. This is it's not the exact lab room that we're using now, but it's the same machine. And so people would come and sit in this kind of type of chair, and they get stimulation with the magnetic coil and the electrical stimulation. I'm putting here also the photo of Dr. Francois Genadri, who's also on the call, who's the fellow who's been leading this work, so he would be one of the individuals, along with a large team like Safar and others in our group, who would be delivering the intervention and would be doing all the assessments and the follow-up with the individual receiving this treatment, this intervention. what we're looking for to complete the study is to enroll 100 individuals with MCI and 50 healthy controls, all the individuals with healthy controls. So this is a summary of the visits that people receive. First, at baseline, the individual gets a clinical and a cognitive assessment to test their memory and their especially working memory. And they get an MRI of the brain because the MRI is needed to locate the frontal cortex with precision to where we're going to simulate. Then we do what we call a baseline assessment of plasticity, of the DLPFC plasticity, or dorsolateral prefrontal cortex plasticity. That's the frontal region plasticity. So we measure the plasticity at baseline. We also deliver the NBAC, which is NBAC is a working memory test. So that's the measurement of the working memory that's linked to frontal region function. And then people are randomized, if they are in the MCI region, they are randomized to receiving either active PAS or PAS control. That's the control intervention for 10 sessions. Again, typically 5 days a week for 2 weeks. And again, here to the right and the left. And then after that, we do the follow-up assessments, doing again immediately 1 and 4 weeks after the interventions. The individual who participate as healthy controls, they don't get the intervention. They just get the baseline assessment. And the idea is to use the data from the healthy controls to compare the results at baseline and after the interventions from the individual with MCI to see how do they compare to people with who are healthy controls. And that's I put here also the email of the study if anyone is interested in learning more about it. I'm gonna stop here. This is again, these are the different funding agencies that contribute to this work, but also all the team members, collaborators, and who I enjoy working with and make this work possible, as well as a large group of trainees who have contributed to all these stages of development of this work. So thank you, and I will stop here.
Thank you so much, Dr. Raji. That's certainly